Thank you, everyone, and it's a pleasure to be here. So this is a project back on Earth and dealing with the practicalities of hospital life and patient care. So I'm going to talk about how this technology enabled a pilot project to get off the ground. So I go straight into the vision, which was, how do we implement personalized medicine across the patient's lifespan using whole genome germline data at the McGill University Hospital cancer patients. And it was clear that to us, that as scientists, that detecting all heritable genetic variants at diagnosis, at the point of diagnosis, has the potential to improve individual care. And these things are known because you can assess the risk, inform the selection of chemotherapies, informing the use of radiotherapy, predicting adverse effects, and also be able to look at the hereditary origins, risks to other common diseases, and understanding the hereditary origins of risk, and inform basically also the future. And the idea was to follow the patients through their lifespan, basically, and inform future care. The idea is nice, but as we found out, it's not easy to implement. So it was led, we formed a project led by cancer geneticist, Patricia Tonin, in Montreal at the McGill University Hospital and Research Institute, which was named the OncoDrive initiative. So it's as an initiative for cancer patient-centered but physician-driven genome-based oncology care program. It's easier said than done, and the task was to convince clinicians that whole genome sequencing of the germline at the point of diagnosis could actually lead to what we're promising in the previous slide. So on the one hand, the task was to improve the clinical management of all cancer patients, and then also to create a biobank and data bank and enable also future research. So the first, after a few years of deliberation, we managed to put a project together, of course, led from the hospital side by Patricia Tonin, from the surgery side by Sarkis Meterisian, who is the director of the breast cancer clinic, and myself from the genomics and data analysis side. So the first task shown in the middle was to develop the whole resource, the biobank, and combine, which is easier said than done, not only data reflecting the entire genomic landscape of the patients, but also integrate this with clinical metrics and cancer family history data. Then we will generate data that hopefully would be clinically actionable. But what we needed to demonstrate was that it was feasible to do all this. It wasn't straightforward, I can tell you. And we want to assess how the patients would be engaged in such a project, how the clinicians would be engaged, and how we can develop a new framework for a biobank and a research resource. So it had to be a combination of a clinical resource and also a research resource. Very ambitious, but then we tried to convince the hospital, and all these efforts, at least in Canada, are from the bottom up, so you need to convince the hospital that this is worth doing, that this patient-centered, physician-driven, genome-based breast cancer program is actually feasible and will have a lot of impact, particularly for cancer risk management and other diseases. So we set up a corresponding, if you want, pipeline that will help further decision-making and research. So it expanded then as a big grouping between the hospital, myself, and then we have an ethics and governance committee, collaborators, advisors, and finally some financial partners, which is very important in this case. So the hospital said, okay, you want to do all this, I'll give you as little money as possible and try to perform miracles with it. The challenge accepted. How can we do this? So we put some money from the foundation of the hospital, the McGill Genome Center, so my own money at the end, and we applied for some support from FIQS. Anyway, what we put together was the following. How do we generate good quality data at the most cost efficient way? So we set up to establish a pipeline using the newly installed DNBSEQ T7 in the lab to have cost-effective, and I'll go into that sort of in good quality sequence, and then we could assess the utility, of using this sequence in routine clinical, data to produce data from germline and interpret it, and then at the end of the day integrate all the germline data with the clinical data, family history data, and create a biobank. All these things did not exist at the point. So that was miracle number one to put together. And then number two was to actually set up a pipeline and have people collaborating with each other. And as you can see here, we set up a whole, Patricia Tonin set up a whole recruitment team shown at the top, an assessment team to take all the data and interact with the patients and the physicians. My job was to produce the data and set up the analysis of the data. And what I like to highlight is that we implemented several data filtering, if you want, steps in order to generate interpretable data. And thankfully, we were aided by this from using reporters like the cancer predisposition sequence reporter and the personalized cancer genome reporters and other tools that we put together in order to generate and compile interpretable information for the clinicians. In first instance, just to explain, in order not to shock them, because when we show the slides of how the data are analyzed to the clinicians, they panicked and they didn't want to know. So we said, okay, we'll start slowly by looking at solid cancer predisposing genes and then move forward. And we also, due to the very little money we received, we focused on breast cancer and on people that were diagnosed with invasive breast cancer. So the technology, was described by Radu, but I'm just showing it here again. So to confirm that we performed we generated the data using PCR-free whole genome sequencing, library prep, and then here is the interesting part from the technical perspective, is that the T7 actually is able to produce at low cost and high quality data from 15 samples per flow cell. So basically you don't need more than 15 samples to start sequencing. And the other thing that was of interest here is that it takes only 24 hours to generate the data. Therefore, we assess that this is quite a reasonable way to implement as a clinical pipeline and be able to sequence small batches of samples, which would be suitable for the pace at which cancers are being seen, patients are being seen at the cancer clinic, but easily scalable from 15 to 60 samples per day. The other thing that was very important is that we performed extensive benchmarking analysis. So we generate a lot of data with our platforms at the Genome Center and compare them to each other. And you can see here what we concluded, I think, from Anyway, it doesn't seem to work very well, but if you look at the table here, it shows basically from top to bottom benchmarking. So we generated sequence from the genome in the bottle trios, and at the top it's the truth set according to the genome in the bottle effort. Under that is the Illumina data using NovaSeq. Below that is the data from the T7, and then all analyzed with a number of different pipelines to make sure that we are not biasing things. And then under that, it's PacBio-NT data and 10x Genomics Linktree data, all generated from the same samples. And what is interesting is that you can, there we go. You can see that actually the T7 performs very well in terms of recovering true positives for both SNPs and indels, so deletions and insertions, and it performs very well compared to all others in terms of false positives, so very few, and also very well in terms of false negatives. So we concluded that this is a good platform to test and bring through its paces using this effort. And pleasing to know that this is combined with very low cost. So just to say, the project would not have been possible with other platforms existing at the time. So the recruitment started in 2021, but then it was interrupted due to COVID. So, and it took, well, the work, just to suffice to say that there were so many obstacles to get approval for this project that it took as long to do the work as it took to get the ethics approved, but we managed. And at the end, 133 patients were consented in first instance, and 102 prioritized as meeting all our criteria. The other thing that is important here and related to the advantages of performing whole genome sequencing is that our patient population is really mixed. There is a background, so Quebec has its own 6 million francophone population, which are all derived from about 8,500 original settlers. So that gives a particular distribution, population-specific distribution of risk alleles. And then also, it varies due to the high immigrant population, like myself, which comes from all over the world, as you can see here. So we need to have whole genome level information of all polymorphisms and variants in order to interpret the risk of any particular predisposing variant. So at the end, we narrowed down to 100, so we sequenced 120 samples just within a few weeks, and then the work started to try and make sense of the data and start convincing clinicians that it's all worth our effort. So I'm going to show you just the pipelines that had to be set up. We managed to tie the clinicians down and so that they didn't run away when they saw this, but the pipelines were already established for SNP calling. We established new pipelines for calling structural variants. And we also generated a database to house all the data, shown on the right. And also the other thing that we did immediately was to generate very quickly in parallel coverage data for all the breast cancer genes of interest and other future cancer genes of interest, to actually assess that if we use the data, that we always have good coverage, meaning 40x type coverage for the genes of interest. Now, the first thing that came out from the analysis when we perform a principal component analysis using all the common polymorphisms was indeed our patients came from all over the world. They're all shown in pink. So you can see that, yes, we had top left, these are people from European descent and would fit with the Quebec population overall, but you can see that we have patients from Africa, from East Asia, and from India, and South Americans as well. So that was already very useful to know. The other thing that we were agonizing about was that without us knowing, there were patients included who actually were already screened for hereditary cancer genes like BRCA1 and BRCA2. So the challenge was, will we find the same mutation as the ones, or variants as the ones identified through the clinical screen? And to our pleasure, all of them were identified, shown here at the top, very nicely, and this result came out very quickly to inform the clinicians. But in addition, we found quite significant predisposing mutations, so pathogenic and likely pathogenic mutations in quite a number of other patients who were not selected or did not meet the strict criteria imposed, at least in Quebec, in terms of who is allowed to go into sequencing. So we recovered quite a few mutations, and as you see here, worryingly, there are BRCA1, BRCA2 mutations, TP53 mutations, ATM, and RAD51C as shown here. So all these were recovered, and to cut a long story short, the analysis overall revealed another 32 pathogenic and likely pathogenic mutations in the, if we look at 433 cancer genes all over the genome. So all this is shown here. So that was one thing that, the clinicians were happy about. We also performed structural variant analysis, and again, a patient was sneaked in who had already a deletion within BRCA1, diagnosed by MLPA, and indeed, we did actually found this. To make sure we combined at least 2 different callers for structural variants, as you can see here, and we immediately recovered this diagnostic mutation as well. Overall, we found quite a number of structural variants in the 433 gene set, in total about 57, covering 16 genes from the set that we analyzed. So that was also very interesting. Then you can ask, well, okay, you found all this. Is there anything else of interest? Can we recover biomarkers? As a proof of principle, we went to, and collected data about biomarkers from CIVIC. And then indeed, we did find some interesting biomarkers shown here. Well, TP53 is known, but in this case, we also found a variant in ERCC2, which indicates cisplatin resistance. So that was already very useful for the clinicians to show. It was not known before as well, and in KIT, and of course, BRCA1 and BRCA2 mutations that allowed these patients now to move straight into PARP inhibitor therapies. What else were the secondary findings? Yes, there were secondary findings. And indeed, we found a patient who is a compound heterozygote in a pathogenic variant in the biotinidase gene causing biotinidase deficiency. So now this patient is being followed up as a result of this work. Things are not as simple, and we have quite a number, as you can appreciate, of variants of unknown significance, and it depends on which database we use. So if we look at ClinVar, for example, it's about just over 100 of these that are recovered, and the number is narrowed down when we use the cancer-predisposing sequence reporter. So these take priority in this case, but then all of them we are sifting through as we speak. So the good thing is that there are about 10 of these that are coming up per patient, and the follow-up is reasonable. It takes a long time to go through this. So if I summarize the data, One thing that came out, which was very important, was that not only did we recover all clinically diagnosed mutations in all the patients, but actually we identified mutations in predisposing variants in patients who have not been screened for one or the other reason. So this was already quite a successful part of the project, but in total, basically, for example, if I summarize here, there were 11 variants that were known, all of them were recovered, 7 were clinically actionable from the new ones, and then we have about 32 that are being investigated for going into in the clinic now for actions in 17 cancer-predisposing genes and structural variants, also probably pathogenic in another 16 genes. And we identified biomarkers, which is good for the clinicians, and secondary finding possible metabolic disorder So I'll stop here in a sense saying in a final conclusion that indeed the findings went into the clinic. Patients are now being followed up. We are looking at other diseases like heart and diabetes, and now interacting with patients and continue the research and writing up the work. Thank you. I'll stop here.